He's based at the Veterinary Sciences Laboratory in Belfast and has a keen interest in all things related to bacterial zoonosis, the genetics and genomics of the pathogens behind them, and the animals hosts they infect. His main interest, however, is bovine tuberculosis caused by Mycobacterium bovis and its evolutionary history and epidemiology in the UK and Ireland. And he's a member of the DERA High Level Scientific Advisory Group and the UK government's Department of Food and Rural Affairs TB Microbiology and Immunology Research Advisory Panel. From a personal side, he's also the best collaborator in the world. So, Adrian, the floor is yours. Thank you.
You say that to all the boys, though, Lily. That's very kind of you. Thank you for the invite. It's a real privilege to be asked by the union to do this, and particularly with just zoonotic TB, just seems to be such an interesting thing for so many people at the minute. I know it's certainly piquing our interest. Our normal bread and butter here is to deal with badgers and cattle, and occasionally deer, but increasingly we're seeing zoonotic cases, which is unfortunate for the people involved. But today, it's a real pleasure. I'll say again, good morning, good evening, afternoon to everyone. The great thing about this union's reach is that it reaches an awful lot of people. And as more and more people come to the webinar, you'll get a feel for just how many people are representation around the world, how many people are here. But today, we're going to hear an interesting talk by two luminaries in the field of TB. We have Professor Ellen Brooks-Pollock and also Dr. Amy Thomas, both from the University of Bristol in the UK. So I'm just going to give you a brief introduction to both of them. Ellen's a professor of infectious disease modelling at Bristol's Medical School. She's focused on using disease dynamics theory and epidemiological data to answer policy questions about transmission and control of infectious disease. And she also has a big interest in developing tools for communicating the findings of these types of models to non-expert modelers, people from a policy background. So communicating science is quite a big deal for her. In the last few years, as you would expect, she's done a lot of zoonotic agent work on things like H5N1, influenza, SARS-CoV-2, the pandemic that we all just lived through. But as well as that, the focus of today's talk, Mycobacterium bovis and zoonoses in humans. And among her extensive publications, I think the first thing of yours I ever read, Ellen, was in 2015, the paper you did with James Wood and Royal Society of Proceedings B, Eliminating Bovine TB in Cattle and Badgers, Insight from a Dynamic Model. And that paper really stuck with me, and I think it's a bit of a seminal one for the bovine TB epidemic in Britain and Ireland, because I think, well, you can keep me right, Ellen, here, but I think you really showed the importance of dealing with TB properly in the cattle population. And I think since that period of time, with other studies and other technologies have come along, I think it's only proved the salience of what you showed in that. So excellent paper if anyone's interested in the cattle and badger TB situation in Britain to read. Moving then on to Ellen's colleague, Dr. Amy Thomas. Amy's a postdoctoral research fellow who's also at the University of Bristol's Medical School. She has a great interest in the carriage and transmission of infectious diseases in livestock and humans, so it comes as absolutely no surprise, of course, that she'll be interested in zoonoses. Very broad skill set, which she applies to zoonotic infections, things like molecular biology, immunology, and epidemiology experience. And her interest in publications have tended to focus on the same pathogens that we described for Ellen, so SARS-CoV-2, influenza, also bovine respiratory diseases, but of course bovine TB and Mycobacterium bovis. And she was the lead author on a really interesting article called High Mycobacterium bovis Exposure but Low IgRA Positivity in UK Farm Workers, which was published last year in Zoonoses in Public Health, and I think probably forms the basis of today's talk. So without any further delay, I really want to welcome Ellen and Amy today, and they're going to talk to us about their work. The talk is entitled Decoupling Mycobacterium bovis Exposure from Infection, Insights from a Study of Latent TB Infection in UK Farmers. We'll have a question and answer session after this, but Ellen, maybe take it away. Thank you.
Thanks so much, Adrian. I like shows how old I am, the fact that I've got papers on bovine TB from over 10 years ago. So I'm going to share my screen, and you can tell me whether it looks okay. Looks okay? Oh, I can't see it. Oh yeah, I can see Adrian. Can you nod, Adrian? Can you see my screen? Yeah, perfect. Great. Well, thank you so much for inviting us to give a talk on one of our favourite topics. Amy and I are based at the University of Bristol, which is in southwest England. My background is in infectious disease modelling, and Amy is really the immunology brains behind everything. And we are going to talk about a study that we've done that really kind of led to more questions than answers, maybe. And we'd really appreciate any thoughts and comments on the results, and also what we're going to do next. So we've got a Menti slide at the end. So what we're planning to do is I am going to give a kind of brief overview of the last 150 years of TB and bovine TB in England, and then introduce the ZooTB study. Then Amy's going to talk about the ZooTB study, and we're going to then talk about hypotheses and future work. So I know lots of you already know this, but it's always interesting to think about TB and what the situation was in the UK and in Europe, because zoonotic TB is often not really considered in low incidence settings. So in the 19th century in Europe and North America, it was estimated that 1 in 4 people died of TB, and about maybe 3/4 of people were infected with TB at the time. Now, at the time, in the 1930s, it was estimated that about 2,500 deaths were due to zoonotic TB in England. And you probably have read the really nice Vinicky and Fine paper in 1997 that estimated the proportion of the risk of infection, including from contaminated milk. So, as you know, the kind of story in currently low incidence settings is this big decline that we saw in human TB cases, starting in the 1900s and declining with treatment and everything else. So at the time, though, 40% of cattle herds were infected with bovine TB. And this is a really nice paper from Susan Jones that mapped bovine TB infected farms. So this shows this is a study that was looking at TB herds that were bringing milk into Manchester. Manchester, just put this little picture on the left, is a city in the north of England. And she mapped farms that were infected. So farms that had TB are a coloured in circle, and empty circles are farms that didn't have TB. So around 40% of all cattle herds had TB at this time, so kind of in the 1900s. So huge rates of infection in cattle. And this was really such a concern that the British government launched this Royal Commission on Tuberculosis in 1901. And this ran for 10 years. It was administered by William Power, who was the chief medical officer at the time. And that was really because the relative importance of contaminated milk versus person-to-person transmission was not known. So the aim of this Royal Commission was to establish whether bovine TB and human TB were the same infection, whether animals and people could infect each other with it, and to establish under what conditions transmission took place. So I always like reading it, because obviously we all think Robert Koch is brilliant, but this is one example of where he got it wrong. So his experiments, he was, as you probably know, he was taking infection from cattle, he was taking human TB and infecting cows, and then looking to see whether he could distinguish the TB due to human TB and the TB due to bovine TB. And he found out that he couldn't distinguish the infection in cattle due to MTB versus M. bovis, and therefore he actually recommended that no measures were introduced to protect against contaminated milk and transmission from cattle to people. Nevertheless, from that, widespread cattle testing was introduced in the UK, and we had an attestation scheme where farms had to get a kind of TB-free status, And this was really massively successful and brought infection rates down really low. So these triangles you can see on this figure are the estimated prevalence of TB reactor cattle. This increase, the early increase probably isn't an increase, but you can see the kind of decrease in the 1960s. And the test and slaughter rates really brought bovine TB in cattle down to very low levels. So you can see that by the 1980s, we have low rates of human TB, low rates of bovine TB. We also have pasteurization, a bit problematic for the British cheese industry, but generally very good in terms of TB transmission. However, at the time, this paper came out in 1974 that was reported TB infection in a pocket of southwest England, in Gloucestershire, where they were investigating the cause of TB in cattle that they hadn't been able to get rid of with test and slaughter alone, and they traced it back to some infected badger sets. So this was really the first indication that badgers were involved in bovine TB transmission as well as cattle. So you probably know the story since then. So we saw the very nice low rates of TB in cattle in 1970s and 80s. This is just number of reactor TB infected with bovine cattle infected with bovine TB in the UK. So you can see that since the 1980s, we've really had a continual increase in the number of cases, and now we're kind of, you could say we're at a pretty stable level, but of this higher rate of TB in farms in Britain. And this map on the right shows the number of reactors per county, and you can see. So it's coloured in. It's not that there are no cases in other parts of the country, but the vast majority of cases are in the southwest of England. And Bristol, I don't know if you can see my mouse, but Bristol is kind of right in the centre of the southwest. So it's the biggest city in southwest England. And it's really the centre of where bovine TB cases are found in cattle. So at the same time, our situation in human TB is that we have fairly lowish rates, although not as low as other low incidence countries, about 6,000 to 7,000 human TB cases notified in England every year. And a high proportion of them, about three-quarters of individuals, are not born in the UK, so have a country of birth that's outside the UK. This map on the right shows where cases are from, so they're predominantly in urban areas, exactly the kind of risk factors that you expect in low incidence settings. Now, at the same time, I moved to Bristol in 2015 when that cattle and badger paper came out, and I started attending TB network, human TB network meetings. And I was very surprised to find that we still had cases of M. bovis in England. So here you can see the number of cases of individuals diagnosed with confirmed M. bovis in the UK since 1999. So since 2010, we've had about 361 people diagnosed with TB due to M. bovis. And the idea is really that some of this is due to infection, very old infections, pre-pasteurization. So, and just one little note. So if you look at the cluster sizes of M. bovis cases in the UK versus MTB cases, you can see that the M. bovis cases tend to be larger than you would expect. So the black dots are the sizes of MTB cases, and the red and the purple dots are M. bovis and M. africanum in the UK. So you tend to get, for the incidence, you tend to get larger clusters of M. bovis. this really nice paper came out by Jennifer Davidson. She was in the TB team at the Health Protection Agency at the time, comparing M. bovis cases to MTB cases in the UK. And she found this absolutely staggering odds ratio, so an odds ratio of about 30 for individuals working in an agricultural or animal-related occupation for M. bovis compared to MTB. And then there are other things in the literature looking at case studies of individuals who have been exposed to bovine TB and then subsequently being diagnosed with M. bovis. And just to say, these M. bovis cases are not your typical TB case in the UK. So they tend to be UK born, older individuals, and with this very high odds ratio associated with working in animal related occupations. So what we really want, obviously the problem with just looking at M. bovis diagnosed cases is you miss this huge, this potentially huge number of individuals who are exposed and infected with bovine TB. So if you think of this kind of inverse pyramid that we always think about in epidemiology, we know the number of individuals that are diagnosed with M. bovis, about 30 a year, but then they are probably, diagnosed and laboratory confirmed. They come from a larger pool of individuals who may be symptomatic and treated for TB, but not confirmed M. bovis. And they would come from an even larger pool of individuals that are infected with M. bovis and exposed to M. bovis. So really the aim of what we were trying to do with the ZooTB study is understand this pool of potentially this larger pool of infected cases, MTB case, M. bovis cases in England. And this is where I'm going to hand over to Amy.
Thanks, Ellen. I've just requested control.
Yeah.
So hopefully that works and I'm able to shuffle the slides.
I just allowed control, so maybe you want to try experiment.
Yeah, I've just tried to advance one.
Yeah.
Not working for me, and I can't see an arrow, so that's not very helpful.
I can see. Okay, shall I just go down? I think I'll be able to. I know what you're going to say.
Yeah, go on, you go for it. Okay, thank you. So I'm going to go through really what we've done with the ZooTB study and the results from it, and then we're going to ask for a little bit of audience participation. So we aim to estimate the prevalence of zoonotic TB in people in the southwest of England who had contact with cattle. And we wanted to determine, if any, what the risk factors are associated with this exposure and do a comparison between MTB and M. bovis transmission potential. So this was really a collaborative project between us at the University of Bristol, a North Bristol Trust team who did the testing for us, UK HSA, so that's Isabel Oliver, and Cambridge, we had James Wood, and also our Animal and Plant Health Agency in providing essential data, which I'll tell you about. So next slide, please. So to start with, and this has really been central to how we've been conducting zoonotic disease research, is the use of public involvement. And before Ellen even acquired the funding from the Wellcome Trust for this study, she started just by kind of gauging the willingness and the study idea with potential participants. And this was really informative of shaping that first grant proposal. And this took the form of, nonetheless, pints at the pub with some local farmers. And this was through a partnership. It was already a pre-organised event through a local large animal practice who were holding an information evening. So we're able to sort of piggyback onto that and do some public involvement there. The funding bid was unsuccessful, and we started, before we did anything, at this kind of study development stage, to again go back to the farming community and to get their knowledge and opinions on our kind of sketched out study design. But this was slightly challenging because we were in COVID at this point. It was 2020 and we were in and out of COVID restrictions. And we would ordinarily have always wanted to have done this face to face. It's worked very well. And, you know, farmers like to generally have a chat. So we put out a call on social media through Twitter, not really knowing what to expect because of kind of anecdotally a varied kind of computer literacy and engagement, let's say, among the farming community. But actually we had some really positive responses, possibly helped through the fact that everybody was having to then communicate through Zoom in one way or another. So we had 4 farmers. 2 were, I would say, they were in the young farming category, so they're kind of around 35 and under. And then we had 2 retired farmers, but still very active. And this was really central to shaping how we were going to do the study, particularly as we had now these COVID restrictions to navigate, and how are we going to get out there and sample people. And one thing that we suggested, and this just shows the value, is that as a researcher, you think it's a good idea, but we said, well, everything's shut down, but the supermarkets are still open. So why don't we go to the supermarket and we could do like a little pop-up testing in the car park? And the farmers all just laughed at us and they said, we do not like going to supermarkets, and that's a really bad idea. And they said, you should consider agricultural shows when things get better. And that's exactly what we did. And it was really formative for them to say that. So we then moved on to doing a more kind of nuanced form of co-development and really working very closely with the farming members. And this was highlighted to us. We had in the session, we were really new to public involvement at this point, by a kind of an expert within Bristol. And she said, they're really a very enthusiastic bunch, and you'd be very silly to not try and do some more work with them. And that's what we did. So on the next slide, we really started to have more in person when we could, and online meetings, and just generally found engagement to be very high. And of course, one of the main benefits is they give you this expert knowledge. And this photograph is Ross Simmons. So he's a dairy farmer in Cornwall, and he attended our session, which we had during one of the COVID restrictions online, and said to us, I'd really like to get involved. So we almost recruited Ross to become kind of a collaborator and a co-researcher on the project. He worked very closely reviewing study design, materials, inputting into the questionnaire, tweaking the questions with us, and interpreting the results. And we paid him for his time. Now, one thing to point out is really challenges with this is your diversity of contributors is often could be quite limited. And we did face some spurious inquiries when we had put incentives to take part, so particularly kind of monetary reimbursement and value, and we wouldn't do that again. We would probably only disclose that information after some screening. So we had individuals who would pose an expression of interest. They looked like, you know, a farmer who we would want to talk to. They would then come into an online meeting, and that's the difficulty again with an online meeting, and not want to turn their camera on, and we would have genuine participants there and having to navigate that. So that's something to just be aware of if there's anyone here who's thinking about doing some public involvement. And I spoke about COVID restrictions. So next slide, please, Ellen. So just to go through the study design in more detail, we asked individuals they needed to be 18 years old, had to have had contact with cattle, and originally we stipulated a minimum of 10 reactors in their herd. They would have no history of TB disease and no TB-like symptoms. So we weren't interested in assessing for individuals with active TB. We really were measuring exposure through latent TB. Now, taking part involved a questionnaire. This took about 10 minutes to complete, and we had across various different sections. So participants gave us information about themselves, their general health status, their vaccination status as well, self-reported, then work and their bovine TB status. So this included their self-reporting of the number of reactors and potentially the number of lesioned animals in their herd, and their daily contact with cattle and farming practices. Now, we uniquely were able to access through collaborators at the Animal Plant Health Agency historical TB test results, and we did this through the linkage with CPH numbers, so the county parish holding number, which is the unique herd number for each herd which will be tested. And we looked at bovine TB exposure in people using the IGRA. So we were looking at latent TB infection. And this was done through colleagues with North Bristol Trust. So we conducted 4 study test days. They were all almost at agricultural shows as per the recommendation of the public contributors. So we started our first one, which was a pilot at the dairy show, and we went back again to the dairy show the following year. So we had 26 people in the first pilot study. We did some improvements or modifications following this period. We went back. We then went to an Agri-fest, which was more of a beef orientated agricultural show. And then to really try to pick up a wider kind of tranche of people who were not necessarily just farmers, but working in other agricultural populations, we put on our own study testing day. And we did this again with public contributors sort of co-developing it. And we went back to this idea of going to a pub, a public house somewhere near Bristol. We did lots of recruitment ahead of the day. We had the phlebotomy there at the pub. We put on a pub lunch. We almost had, I think, 20 people lined up to come. But in the end, we had about 5 who did the questionnaire and did the sample, because maybe in hindsight, it was just a risky period. You'll see it's kind of springtime. It's around lambing. And we were really trying to get some more vets and veterinary professionals who said they were coming, really, really keen on coming. And then obviously your workload just changes at the last minute. So we probably would rethink that if we did it again. So a little bit on the reflections from our pilot. I mentioned that we had this minimum criteria of 10 reactors, which when we were going, we were kind of actively recruiting at the show. So people had lots of time to spare to have a chat with us. And then we would say, oh, do you have 10 reactors? And they say, oh, no, I don't think I have. So we found this was actually maybe a bit too stringent. And then the other thing, we were missing people who didn't work on a permanent holding. So this was vets, for example. So we made some modifications and we just said active involvement with TB cattle. And this opened us up to a much wider range of professions who were still potentially exposed to TB infected cattle. We found that once we'd got through the initial scoping and consent, we whizzed through the questionnaire. And they say, oh, is that it? Oh, well, I'd love to tell you about this and I'd love to tell you about that. So there was very high engagement and an appetite for more questions. So we put a few more in, which try to capture this non-farming demographic. And we asked a little bit more about perceptions of zoonotic risk. And at this point, we filed for some extra ethics to collect an additional blood sample because, we were thinking about serology. So in total, we recruited 90 people. We had 76 farmers, 10 vets and 4 in kind of other related agricultural occupations that still had contact. And they were, the median age was around 45, majority, so 60% odd were male. Almost everybody was white ethnicity. Little over half, so 57% were on dairy farms, and the same had worked in the role for over 20 years. So this was really fitting with a UK kind of scope on the farming cohort. And we then categorised into these risk categories for exposure, and I'm going to tell you a little bit more about them. So I mentioned that we did some data linkage with the recorded TB, bovine TB data for the herds, for which individuals consented to give us their CPH numbers. So this shows the test year over which we had data linked for and the number there. And so you can see really the majority were in the last 10 years. And this was really fitting with the fact that we felt we would take forward this chunk of data because it would most likely be more reflective of the natural history that we were trying to explore. And in terms of the herd sizes in this lot, so just considering the last 10 years of testing data, you can see quite a range, but mostly we didn't find more than 1,000, and really the median somewhere around this 400 mark overall. So quite a range of sizes, but quite centred. Now, when we look at the this figure on the left showed the number of herd reactors against the number which had lesions at slaughter. So we generally see that the more reactors you have in your herd, the greater proportion of those are going to have lesions at slaughter. And we were really interrogating this to try and get an understanding of what we thought the exposure level might be in these people. And we also asked about contact hours. Now, of course, you can see here on the right figure, this is the number of hours a day that individuals reported having contact with cattle. So it goes from 1 right the way up to, well, mostly 12, and then somebody way out over here, which is, around 15 hours. Now, most people are sort of having contact between 1 and 6 hours a day, but we didn't really find a trend between the total number of reactors and the number of contact animals and contact hours. We saw, we really felt that there's probably a high level of self-reporting bias. Everybody thinks that they're doing, they probably are working all the time, and it's really hard to actually quantify that level that you're really having the contact with animals. So we then categorized into these risk groups for which we then did our exposure analysis on. Now, before I go into them, because we had the linked APHA data on the reactors and the lesions, we did a correlation to this on the self-reporting. And we found, in general, the number of reactors was reasonably well correlated, but much less so for the number of lesions. And generally, for both measures, individuals are much, they're underestimating the number of reactors, and particularly the number of reactors with lesions in their herd. So using just the APHA data, we categorize individuals into these risk categories based on the median number of reactors they had in their herd. So that was 36. So if they had less than 36 reactors in the herd, they were in our low risk group. If they were between 36 and 85, they were our medium risk, and anything over 85 put them into their high risk group. So what did we find based on the risk group? Well, we didn't see a difference between demographic factors among these exposure groups. We had just to add on the far right column of this table, these are individuals. We had 17 who couldn't be linked. So there's no CPH number. Really a high number. Overall, it was 67% reported having had the BCG vaccine. And we saw, so as you would expect, given our grouping here, if we look at number of reactors, this is increasing with the risk group. And again, we saw increasing number of lesions present with the increasing risk groups. Now, really quite a high percentage reported consuming raw milk frequently. And this was particularly more marked in the medium, so around 94% in the medium risk group and 76% in the high risk group, and slightly lower, 50% in the low risk group, and even lower in the no CPH group. And interestingly, the low risk group found themselves to be at risk, 70% found themselves to be at risk of TB. Everybody in the no CPH group found themselves to be at risk of TB, but those in the medium and the high risk groups found themselves to be at a lower risk. And again, with the contact with daily contact with cattle was really reported to be high across all risk categories. And as you might expect, it was lower with the no CPH. So these are individuals who generally didn't have maybe a CPH number. So perhaps they were working in an occupation that wasn't quite as full with contact hours. So these are our IGRA results. Now they're split here on the left with the peptides. I mean, many of you will be very familiar with IGRA. So there's ESAT6 and CFP10 for CD4 cells. Then this again, but with an addition of some new peptides, which are undisclosed, as for CD4 and CD8. And then we can isolate the effect of the CD8 by doing the difference between them. Now, we had one individual who was really unequivocally positive, which you can see in the yellow. And we did have a look at two different thresholds. So anything above 0.35, this is a test standard, would be considered to be positive. And then we have a look at a more stringent criteria as per publication by Harada in 2008. and this was 0.15, but still all of our negatives were really below this more stringent threshold, which would increase the sensitivity with a slight decrease in the specificity. Now, the next slide really just shows the same results, but in a slightly different format. So you see the TB1 antigen on the left and TB2 on the right, and this is now the number of participants with the level on the x-axis of interferon gamma being produced. So, you can really see the majority, somewhat 76 people clustering right around 0, and the triangle here for the one positive individual. So overall, our IGA positivity was 1.1 up to 6.9 if you consider the confidence interval. And this individual was in the high exposure group that we had categorized them into. So this gave an IGA positivity in the high exposure group of 4%, somewhere between 0.21 and 22%. So this is where we, maybe everybody can start to scan the Menti, and we wanted to pose some maybe hypotheses for these results and get your feedback and in response onto what you think might be happening. So you can vote for more than one, and we've got 4 to pose to you. And these are bovine TB in cattle is controlled by the test and slaughter. So, in Great Britain, we test all of them, and all reactors are slaughtered. So we're not really seeing any cattle who are potentially infectious to people in our herds. We found maybe that this was just the wrong cohort and that the human exposure was too low and relating to transmission. Uncertain latency for M. bovis. So this is the third one. We were taking the assumption that M. bovis natural history in people is similar to M. tuberculosis natural history in people, and that may not be the case. And then alternative diagnostics. Was the IGRA the appropriate test given this maybe uncertainty in the natural history? So maybe we'll give you a couple of minutes and gather some more responses, and then we can have a discussion on this. It's great that you've all managed to find it. That was our main concern, that it was just going to be Ellen and I voting. So you can vote for more than one. Maybe we'll finish so that we get up to 38. And somebody else has just come. Okay, should we move on to the next slide, Ellen? And you can obviously all keep voting for your favourite hypotheses. But it looks like alternative diagnostics needed is winning.
I'm not sure, it might just stop actually if I go on to the next slide, but I could do that. It's interesting that there's not a clear winner, or there's not a clear, because we have been kind of backwards and forwards about what we think. As Amy said, the sample size calculations that we did for this study were based on this idea that for human TB, there's a high number of individuals will develop a latent infection and never go on to get disease. And so I think we'd be very interested in people's opinions about whether that happens for M. bovis as well, or whether there's just a much shorter latent period, or maybe much shorter, or it's much less common to get a latent infection for M. bovis compared to MTB.
Yeah, and we'd also thought maybe that the exposure, with the human exposure too low, that we've not focused enough on the high burden farms, and that even if we have, that exposure doesn't necessarily lead to a measurable T cell response that we're getting with the IGRA. So this was really part of the motivation for taking some extra serum samples for some potential serology analyses later. And IGRA reversion is another item that we've had some discussion on. So in summary, we've really reported this high exposure phenotype of individuals to M. bovis, either through consumption, potentially a potential through raw milk, and direct contact. So really quite high contact rates between TB infected cattle and people, though low IGRA positivity, no clear evidence of false negatives, but as with always, there's some limitation. So I mentioned this high vaccination rate of 67%. Perhaps it's possible that the BCG is maybe promoting early clearance before we even see an adaptive immune response developing, and that could also depend to some extent on the exposure type and the age of the individual. We focused on latent TB. We excluded any active TB, so we're potentially underestimating zoonotic TB prevalence. And we have this kind of imprecise exploration of the risk profile because we're measuring TB exposure over these different timescales. So the number of reactors, the raw milk consumed, the awareness of bovine TB is all happening at different times. And future work really ought to try to bring those together in some way. So finally, this is our website, and you can take a link to the QR code to read the paper further. We have also, just on the public involvement side of things, we'll have a paper out, hopefully a preprint out shortly, and that will go onto the Zoonotic Disease website, and that will really detail how we've been using public involvement with the farming community. And there's some nice infographics that a very good medical student did with us, Genevieve Clapp, on summarising all of these studies in one place. So thank you, everybody, for listening to us. I'm looking forward to the discussion.
Oh, thanks, Ellen. I mean, that was really interesting. And yeah, I was surprised as well not to see a clear winner in your poll. I guess, like, I'm not the greatest immunologist in the world, but I would kind of, on the positive side, I think, I'll caveat this with not having great immunological knowledge of the IGRA test, but if I kind of want to think that what you're showing there is that actually the test and slaughter scheme is maybe not fantastic at getting rid of cattle with infection, as the GB data would show, but it seems to be potentially quite good from a public health perspective and maybe preventing animals in getting to the stage where they're able to shed and at large enough numbers to actually potentially infect farmers. I mean, does that seem a reasonable hypothesis? Is that?
Yeah, I mean, that's the assumption we were working under, that really we're just removing that large burden of infectious cattle, or maybe we're not measuring transmission correctly with the IGRA is the alternative. It's maybe the wrong tool for the job in this setting. There's been some other work in Mexico, for example, which really reported a very high rate of latent but also active TB in dairy farm workers. So we know that it works in certain settings, which there's shared antigens between M. bovis and MTB. It should work. So it raises the question, really, what's happening in this low incident setting.
Yeah, I mean, do you have any the actual cases that you did? You elegantly showed there. Ellen showed them at the start of the fact that you do have these cases which are definite positive TB. I mean, do they have any sort of characteristics that would suggest? I mean, are they found in areas that would have a higher prevalence of TB or a linkage to herds which have had large breakdowns, things like that?
I mean, the trawling questionnaire does ask, so they have this very high. There's a very high odds ratio that they work in an agricultural setting, but it's not linked to the bovine TB data in cattle.
Okay.
My feeling I mean, we would really like to um, kind of focus in on the individuals who have very high levels of bovine TB in their herd, because maybe you're right that the test and slaughter scheme is reducing the overall public health risk, but some individuals, you know, they still have hundreds of cattle in their herds that are reactors with visible lesions at slaughter. And you have to think that visible lesions at slaughter, and if you're drinking the milk, there is exposure there.
You think so? Yeah.
Yeah. So, but I do think, you know, we would be really up for maybe focusing, doing a, it's tricky testing people, but doing something where you focus on these very highly exposed individuals. And then I'm also kind of leaning towards the idea that there isn't a latent state in the same way. So natural history of M. bovis is different to the natural history of MTB. But we don't really have any evidence for it. But it would make sense in terms of these findings, because if people rapidly move to active disease, then you wouldn't be able to find, there isn't this big pool of infection to look for like there is for human TB.
Yeah, I mean, there is that sort of fascinating sort of host tropism between the members of the M. tuberculosis complex, which would suggest there's something different going on whenever bovis infects humans. It seems to result in different types of pathology. So yeah, there could well be something to that. I mean, does anyone else want to chip in here? I'm saying, do we have to go to the chat for that? Is that the plan? Let me just check.
I mean, just with regards to this latent period, we would be interested to hear about other tests or other diagnostics that might try and get at levels of exposure. For example, Amy mentioned that for our later individuals, we have got a stored blood sample for these individuals, and so there is the opportunity there to develop alternative diagnostics to really try and explore any other markers of infection.
Yeah, well, if we've any immunologists on the call, you can maybe enlighten us on that. That would be interesting. I find this like, Amy, your contrast actually with the situation in Mexico, where there's consumption of things like unpasteurized milk and there's a lot of active TB occurring. And I think even just the contrast of that suggests that your initial hypothesis probably is right, that there's something about the UK's test and slaughter scheme, whilst it's suboptimal controlling disease, does seem to be having, reducing the burden of the potential zoonotic problem. And I'm reminded of it's many years ago now, but the two Torgerson brothers published a paper basically saying a few years back saying, why are we bothering controlling bovine TB? It's really only an economic disease. And there was a raft of kind of replies to them saying, don't get rid of the cat just because you haven't seen any mice. And for me, you're kind of your paper does suggest that that's what's happening. And I think the counterpoint of what's happening in Mexico, kind of brings that home that it may well be true.
I think that's right, but I think it is risky from a public health perspective if you let TB rates go up to what they used to be in cattle. If 40% of cattle were infected with TB, I don't know that we would have the same low incidence in humans. Amy, shall I read some of the questions? There's a few questions in the chat.
I do, sorry.
One of them is there effects of BCG vaccination on your population? Do you know in your data what the correlation between BCG vaccination and individual behaviour, attitude, perception of M. bovis infection risk is, e.g. consumption of raw milk?
That's an interesting point. We haven't really looked at that in detail. I mean, anybody can, because all the data is freely available, and perhaps maybe we'll go away and have a little look and we could get back to you, because there's not a huge amount of variation in those who reported having BCG vaccination, but yeah, we can have a look. We haven't formally done that.
I don't know what you think, Amy. My feeling is that there won't be a correlation between BCG and individual behaviour, because a lot of people didn't think they were at risk anyway. So if they don't consider themselves to be at risk, I wouldn't imagine that would then affect affect their behaviour? What do you think?
No, and I think it's also about the BCG vaccination in general. So the history on that, you know, you're usually given at around 11 years of age. There's not really much time generally to have developed a hesitancy around getting vaccinated. The policy changed in 2013, so it's actually a new cohort of young farmers who are coming through who won't have been vaccinated. And that's kind of driven a separate line of inquiry for us, really. Are they the ones who are maybe, our targeted kind of group to be doing work with because they haven't been vaccinated? And, if that's at all the case, it could be maybe at more risk. So, yeah, I mean, I agree, probably isn't going to be much of a correlation, but we could have a look.
Yeah. Okay, the next question is, does IGRA differentiate between MTB and MTC, so human TB? Could farmers have gotten TB from other people?
So we should have screened everybody out. We did a pre-screening questionnaire, and this I'll answer the next question a bit about screening health workers in touching for caution, and do we think we had an equal positive, basically how we should treat the positives. So this all came out in the ethics application, and we had to do a few amendments, which specifically said if you've got any history of TB, human TB disease in your family, you can't take part. And if you have any current symptoms, you can't take part, because we really didn't want to have anybody with active TB taking part in the study. So we've screened them out. So we shouldn't have any farmers who've gotten TB from people. And then in terms of what was the kind of the route, if somebody was a positive, what would we do? And equally, we had to, if somebody was borderline, what would we do? So we actually had a reporting protocol. If you were anybody who took part and had a result, they'd get relayed that result, regardless of what it was. If you were negative, you just get told it was negative. If you were borderline and positive, you would get the same kind of information. And this was information relating to TB symptoms, what to look out for in terms of if it was to turn into an active TB case. And you had to consent to take part, to give your GP details. And we also notified the GP of positive cases. So this was really on record, and they were to then be aware if any symptoms of active TB came up, they should think straight away it's TB. So that should cover that one.
Sorry, John came in there saying I really think that you need to focus on a more advanced test to look at all those who've had BCG and have lots of cattle infected and none infected. Mm-hmm. So kind of almost like a sort of a case control sort of approach. I think certainly the fact that you found your one IGRA positive in the high risk group, and you've kind of alluded to yourself, maybe it's to go back and look at the more high risk groups would be a better way, perhaps, with hindsight.
Yeah, maybe, and this stratification on age as well, given BCG vaccination policy changing, and that was something really kind of on our mind in terms of the potential for policy changes would maybe be age focused.
Thomas Holder, we use tuberculins PPD minus PPD-B minus PPD-A to bovine IGRA as standard. EC peptides are less sensitive than tuberculins in cattle. Not sure if we know what the case of them is in humans, though. There's an interesting comparison just in from Annabelle Rodriguez. So we just finished testing 250 dairy farm workers in Texas. Preliminary results, 10% latent TB. Used IGRA, quantiferon-2, statistically significant predictors of IGRA positivity, that's P less than 0.05, were number one, working in an enclosed or poorly ventilated area of the farm. Number two, reported that they cannot take time off work to attend medical appointments. Number three, living with other dairy farm workers. We'd love to compare our results. We also collected occupational exposure, so I think we have a collaboration in the offing here, folks, with Texas.
Yeah, that sounds great. We also had some rough questions in there about, do you mostly work indoors, outdoors? We tried to put in some questions that maybe captured aerosol generating procedures, and really this occupational group. Yeah, that would be great.
John Kennebeck has another question. Has BCG being used changed immunologically over the years?
I don't think so. Ellen, do you? I think it's the same. It has always been the same.
Mm-hmm. But there's definitely, as you say, maybe there's a cohort coming through in the UK now who've just never had a BCG vaccination, who in theory should be at greater risk, you'd think. So it's interesting.
Yeah, and interesting from a kind of immunologically naive backbone.
Well, of course, sorry, Alan, go ahead.
Oh, no, sorry to interrupt. I was just going to say it would be interesting we discussed comparing with Ireland as well, seeing as you have a pretty similar bovine TB situation.
Oh, ours is much worse.
Oh, okay. Well, maybe in the southwest.
There are, we were starting to look at zoonotic cases, and we have one anecdotal case, a particularly bad one, that there was a particularly explosive breakdown in a herd, which just huge numbers of lesions and animals, which makes you anecdotally think, is the severity of disease the thing that's key here? And if the test and slaughter regime breaks down in some way that doesn't detect those quick enough, is that when you start to see these things? So we should chat, because we have public health colleagues here in Northern Ireland who are interested in that. And I know that the public health authorities in the Republic as well are interested. I think out of all the regions of the UK and Ireland, we're the ones who've seen the biggest rise, certainly in zoonotic rates. So it would be good to talk. Liliana's come back with one. Apologies if I missed this, but were you able to disentangle exposure due to proximity to cattle versus drinking unpasteurized milk? Did you have farmers with both types of exposure?
Yes, because we categorise based on the number of reactors in the herd. If you remember back to that table with the low, medium, high risk exposure on reactor and then no CPH, the raw milk consumption, having frequently reported it, was very high, 94% in the medium risk, slightly less, 76 in the high risk group, then less in the low risk group, and even less in the no CPH. So no linkage to the number of reactors through their TB testing history.
Yep, I think we could probably wrap up at this stage. Everyone, this has been recorded, and I'm told that any of your questions which come through in the chat after this, we'll endeavour to come back to you. Questions will asked under Amy and Ellen's eye, just to see if we can get an answer for you. But for now, I think we'll call this a day. Thank you very much for attending, all of you. It's really nice to see the TB community coming together to attend this meeting. Thank you to Ellen and Amy for giving us a really stimulating talk, and probably stimulating more conversation than into what exactly is going on. And there's food for thought here for collaboration with other people, particularly that group in Texas. And if you want to do something in Ireland, give us a shout. We could see if we could do something. But everyone, thank you very much. Have a very good rest of your day. Take care, and speak again soon.
Thanks, everyone. Bye.
Goodbye.
Thanks a lot. Thanks, everyone.